BSc, 3rd semester, University of Mysore, session 36, fertilization, details of process with reference to sea urchin. Fertilization, details process with reference to sea urchin. Here the students learn about fertilization, details of process with reference to sea urchin. They learn about the activation of the egg, they learn about the penetration of the egg, and they learn about the reaction of the egg and amphimixis. Students are able to learn about the fertilization details with reference to sea urchin by learning the activation of the egg, penetration of the egg, reaction of the egg and amphimixis. In the previous slide you have studied about the 3 steps involved in the process of fertilization in sea urchin. Now we will go to the next process, that is the activation of the egg, the penetration, the reaction of the egg and amphimixis. So this is what is called as an acrosomal reaction, that is the activation of the egg. So in the activation reaction, the egg membrane is usually surrounded by the egg and the sperm has to produce or it has to penetrate through this membrane before it can reach the egg. So the mechanism of penetration is chemical, and the spermatozoa produces substances of an enzyme nature known under the general name the sperm lysine. So this sperm lysine dissolves the egg membrane locally and make the path clear for the spermatozoa to reach the surface of the egg. And these sperm lysines are produced by the acrosomes of the sperm. But in some mammals the situation is complicated because the presence of a layer of the follicle cells called as zona pellucida around the egg membrane. So it has been found that mammalian sperm produces an enzyme ironidase and which dissolves the ionic acid, and these substances act as a cementing the follicle cells together. And these spermatozoa are unable to release the ironidase until they have undergo some form of physiological changes which normally occur in the female genital tract and which is known as capacitation. But the peripheral parts of the acrosome collapses and releases the lysine, and the central part of the acrosome it elongates and becomes transformed into long thin filament of tube and produces forward. You can see there is a long thin filament or a tube which extends filamental tube, and this extends and become transformed into long thin filament tubes and protrude forward from the end of the sperm, and the structure is called as acrosomal filament. And this acrosomal filament, it is pushed through the jelly where the vital end membrane, the pathway for being cleared by the action of the lysins, and then the tip of the acrosomal filament touches the surface of the plasma membrane of the egg cytoplasm. So these are the events that you see in the acrosomal reactions. Then the reaction of the egg. So immediately after the acrosomal filament of the spermatozoa touches the surface of the egg, the cytoplasm of the egg, it bulges forward at the point of contact and producing a process of iron cytoplasm called as the fertilization cone. And the fertilization cone may be in the form of more or less simple conical protrusion, or it may consisting of several irregular pseudopodia like process. And this fertilization cone gradually engulfs the spermatozoon and then begins to retract, carrying the spermatozoon inward and surrounding by the cytoplasm of the fertilization cone. And the spermatozoa penetrates into the interior of the egg, and the egg shows the sign that is it has been activated. And then the egg in the surface of the egg cytoplasm, which is known under the general name the cortical reaction. And this cortical reactions may differ quite considerably from one animal to another animal, and it is extensively studied in sea urchin egg, that is the formation of the fertilization membrane is first visible changes, and this membrane is suddenly lifted up from the surface of the egg. Simultaneously the cortical granules of the egg break down and disappear, and it is believed that the product of disorganized cortical granules and incorporated in the formation of the fertilization membrane and a vitelline membrane and a side of plasma membrane which surrounds the unfertilized egg. And this vitelline membrane begins to be lifted up from the egg surface because of disorganized cortical granules accumulate in the space between the egg surface and the vitelline membrane, that is perivitelline space. And this fertilization membrane in reality is thickened vitelline membrane, and the thickness is increased from 100 Armstrong to 500 Armstrong, that help in lifting up of the fertilization membrane, which is a defensive reaction, and which prevents the penetration of the additional sperms. You know that only one sperm penetrates into the egg in most of the animals. Fertilization performed by the penetration of the sperm only is called as only one sperm. As I told you, it is called as a monospermy. If there is more than one sperm establishes contact and penetrated to the egg, and it is known as polyspermy. So polyspermy, which is seen in some of the molluscans, tunicates, echinoderms, etc. And this there are two types, as I have explained, as I will be explaining in the next session, that is pathological and physiological polyspermy. And you can also see the fertilization cone, which looks like a pseudopodia like process. And another important step is the nuclear fusion, which is otherwise called as amphimixis. So what is meant by amphimixis means in many of the animals, notably in the mammals, the whole of the spermatozoa with the tail enters into the egg, and only after some time the tail gets degenerated. But in some animals like echinoderms, however, the tail of the sperms break off and left outside the egg. So only the nucleus with the centrioles and sometimes with the middle piece enters into the egg. So when the sperm first penetrates into the egg cytoplasm, it moves with the acrosome at its front. Soon the rotation of the sperm head by 180 degrees is absorbed in many of the animals, and as a result the centrioles coming ahead of the nucleus. So here the nucleus is referred to as the male pronucleus, which starts swelling. As the sperm head moves inwards, it may be accompanied by some pigment cortical cytoplasm, you can see in some of the animals, and depending upon the mature nature of the egg nucleus, the next path is determined, and the second meiotic division, it will be completed only after the fertilization. So after the egg throws off both the polar bodies, the male and the female pronucleus shows the movement towards each other, which is referred as the copulation path. And the copulation path of the sperm and the egg pronuclei are generally not straight, and it will be slightly curved. So the actual fusion of the two pronuclei, that is male and the female pronuclei, may differ in different animals and may be of two types. And in some of the animals like sea urchin and the vertebrates, the two pronuclei immediately fuse together into one fusion nucleus, or it is also called a synkaryon, which is surrounded by the common nuclear membrane. And in other cases like annelids, ascaris and few molluscans, the pronuclei do not fuse as such, but the nuclear membrane in both dissolve and the chromosomes arrange in the equatorial plate. attached to the spindle fibers. And after the completion of the first division of the fertilized egg, that is the cleavage, the paternal and the maternal chromosomes become enclosed by the common nuclear membrane in the nuclei of the two daughter cells. So this is what happens in the nuclear fusion, that is what is called as amphimixis. The last step is, what are the metabolic and other changes resulting from the fertilization? So immediately after the fertilization, many metabolic changes are absorbed in the egg. The important metabolic changes are: oxygen consumption is highly increased, heat production increases, the breakdown of the protein, and ammonia excretion increased, indicating the high rate of protein metabolism, and it is able to affect the protein synthesis. Multiple choice question. Sperm lysine, the enzymatic substances produced by the sperms in mammals, is known as crinogamone, hyaluronic acid, androgamone, hyaluronidase. The answer is hyaluronidase. After the sperm has penetrated an ovum in the process of fertilization, the entry further sperm is prevented by condensation of the yolk, development of vitelline membrane, formation of fertilization membrane, development of pigment coat. The answer is formation of the fertilization membrane. In the process of fertilization, this is true. Entry of sperm activates the egg for completing meiosis. Only one sperm reaches the egg and enters it. Only the acrosome of the sperm enters the egg. Two haploid nuclei fuses and immediately divide to produce two nuclei, which are again haploid. The answer is entry of the sperm activates the egg for completing the meiosis. The fertilization membrane is formed by the fusion of the cortical granules with the inner surface of the membrane, the fusion of the cortical granules with the outer surface of the membrane, the fusion of the inner surface of the membrane with the outer surface of the membrane, the cortex of the cytoplasm. The answer is the fusion of the cortical granules with the inner surface of the membrane. Fertilization cone is formed during the meeting of the gametes, the penetration of the sperm, the activation of the sperm, the amphimixis. Answer is the activation of the egg. These are few references. Thank you.